I really appreciate because it's going to be a very nice session here today. So I'm going to start right now. It's on cell cycle and cell division control, and our first talk is going to be by Michael Dorrity from Stanley Field's lab in Seattle.
Cool. All right, thanks for showing up, everyone. You guys are true heroes for coming us early. So today I'm going to talk about our work in using technology to identify the molecular mechanisms of evolution, and specifically how certain traits might be constrained by protein structure and function. So at this conference, it seems possible that there are some non-yeast people here. So I'm going to review my traits here a little bit, but probably everyone's familiar. They're mating and invasion. So mating occurs in yeast when pheromones are detected between two haploid cells. They project towards each other until they get to an eventual, almost poetic fusion of their genomes. And you can see that here. I'm going to try out the pointer functionality. All right. You can see that these cells are fused, and you kind of have this one with this one here, and you get a zygote. And if we delete genes that are important for mating, these cells are sterile and they do not mate. So also, yeast can invade, which is similarly a kind of readjustment of the cell wall in a way to project themselves and grow filamentously into tissues. And it's this exact process that's responsible for some of the great costs of fungal pathogens, as in human health, and actually to a much larger degree in agriculture. So these traits have things in common, and we know quite a bit about them. So this is the pathway, or rather a rough summary of the pathway that's involved for both mating and invasion. So when this pheromone is sent, it's at a G protein coupled receptor. This eventually sends a signal down through a kinase cascade, which eventually lands on sterol 12, the transcription factor that's most important for binding upstream of mating genes and activating them. And similarly, when invasive growth is induced, a slightly overlapping set of kinases is used, and again, sterol 12 is phosphorylated and goes and binds upstream of invasion genes. So this brought on a very obvious question of how is specificity achieved if you've got this same transcription factor controlling both mating and invasion, things you don't necessarily want to be doing at the same time. And so a lot of this has been attributed to this differential phosphorylation on sterol 12 and how that might change its conformation, and specifically that during invasion, a cofactor called TEC1 is upregulated and binds as a heterodimer with sterol 12, thereby changing its conformation and allows it to bind a subset of the total sterol 12 binding sites, those used for invasion. And so what's interesting about these two traits in this pathway is that the link between mating and invasion is itself conserved among fungal lineages. So we know in plant pathogens and things like Cryptococcus that these same genes are involved for, again, both of these processes. And that in something like Cryptococcus, this is something that's extremely virulent and invades very, very well, however, has very, very poor mating ability when grown at the temperature of us, its host. And so I'm interested in how these things are separated on a more mechanistic level. And so I decided to use a technology we've developed in the lab, deep mutational scanning, on the DNA binding domain of sterol 12 to see how specificity is achieved through specifically this factor. Okay, and so here's a little background on sterol 12. It's a transcription factor that binds as a homeodomain. And it was this paper from decades ago from Stan showed that it indeed looks a lot like a homeodomain, a transcription factor which has basically 3 helices and one which is large and longer and lands in the major groove and makes base pair contacts. And so you can see in this alignment, which was done at the time with several of the other homeodomains that were known from flies, you can see that sterol 12 is actually kind of odd. It has a couple of strange features. Notably, this large string of amino acids between the second and third helix. And another thing that you can't actually see here, which is that the sterol 12 homeodomain actually extends beyond the canonical 60 amino acid DNA binding domain. But it's definitely a homeodomain, as Stan pointed out here. This tryptophan and phenylalanine residue is extremely conserved across all lineages. And so this is very likely adopting some sort of homeodomain-like structure. And so similarly, we know what sequence sterol-12 likes to bind when we induce with alpha factor pheromone. We know from CHIP studies from several groups that this is the motif that sterol-12 likes to bind. And also, we know that if we induce cells for invasion rather than mating, that motif shifts slightly. So these are all things we know going in. So I decided to make many, many mutations in sterol-12's DNA binding domain and developed selections for both mating and invasion to try to disentangle the importance of the binding domain for each trait. So I took a strain which was knocked out for its native chromosomal copy of sterol-12 and complemented that with a library of many, many thousands of variants of sterol-12 mutants driven by their own promoter. And so in each case, we have a culture here, and I'll talk through the mating selection first. So this library is grown up and then mated against a completely wild-type set of alpha cells. And these are plated on diploid selective plates. And I scrape the cells off of those diploid plates and collect the cells from that input library here, and use high-throughput sequencing on both to determine the frequencies of all genotypes before and after selection. And so you can use that change in frequency to infer some function. So we've linked mating to the frequency of, say, this red variant, and if it decreases after selection, we would say that's a poor mater. So similarly, I designed a selection for invasion, which admittedly took quite a long time to optimize. And so again, I start with this input culture, and I plate these on many, many plates and let them grow a couple of days and then wash off those plates. And what you end up with, and this is kind of a zoom in, is all these colonies that have differential invasion. And so what I did next was take all of those plates, kind of cut the agar pucks out and throw them in a blender. And here a lot of the optimization was realizing that the chopped salsa setting is a lot better than the puree setting for the downstream filtering. And so again, we have this selection which tells us which sterol-12 variants are functional, but now for invasion rather than mating. So I'm going to jump right into the data, first with mating. So this is what our data look like. So this is the portion of the DNA binding domain we mutagenized. And you can see here the wild type sequence, as well as a predicted secondary structure. And so we get many, many mutations, and these are just the single mutations showing that for each position of the DNA binding domain, what happens when you substitute an aspartate. And you can see in this kind of former half of the domain that almost every mutation you make is deleterious for mating, though this aspartate substitution is tolerated, say, at this position. And so what's really valuable is we actually have quite a full map, which allows us to view this trait of mating through this set of small biochemical changes and to infer what's important about this domain. And so we can remind ourselves that we're indeed working in a DNA binding domain by looking at, say, these positively charged lysines and see that almost no mutation is tolerated here save an arginine, which would be quite similar at that position. And I should note that the wild type bases or residues are shown with a gray circle, and ones that we have no data are crossed out there. And so one way we like to summarize this data and look at sensitivity of each position to mutation is just look at the mean of all mutations tested for a given site. And so you can see here that some of those lysines I pointed out are among the most deleterious. And importantly, this really, really conserved tryptophan allows essentially no other mutations at that site. And so I'll point out that this assay, we did it over several replicates, and it's very reproducible for individual variants. And specifically with these positional means, we have extremely robust scores that we can infer. And so for the rest of the talk, I'm going to talk about these positional sensitivities to mutation. So here I've got a plot showing the very same thing we were just looking at. It's these mating scores. And as I told you, I did this for invasion as well, but I'm not going to show you the heat map. I'm just going to show you this plot of positional sensitivities. And so it's not very easy to see what's happening here, but you can see it looks like that this tryptophan is also down for invasion. And so what we wanted to do was look at the positional sensitivity of each position for each trait. And this was our expectation, which was, since this is a DNA binding domain, if you mutate some residue, it's not going to be able to bind DNA, so it shouldn't be able to mate or invade. And so what we actually saw varied a little bit, and I guess I should point out that if you were way over here, this would be a strong invasion ability, but without the ability to mate, and vice versa in these bars. And what we actually saw was a little bit different. We saw that our deviation from the expected here was strong, and basically the only thing that behaved as we expected was this really conserved tryptophan, so it's poor for both mating and invasion. And what it looked like is the rest of these residues seem to be almost inversely correlated, and this gets even stronger when we induce invasion by limiting the glucose in the media. And so after looking at this, we thought, oh, this sort of looks like a Pareto front, which is, in this case, just an orange line, but it's a way for engineers actually to optimize, to guide optimization of certain processes, where you have some sort of trade-off to make, and this is the set of most efficient decisions you can make on that. And so we think that this indicates that there's a kind of trade-off between mating and invasion in this domain. We also know that the ones that show the strongest opposite trend themselves tend to cluster in the domain. So they're all from this short segment. So almost every residue here behaves oppositely for mating and invasion, indicating that some parts of this DNA binding domain might be more important for one trait rather than the other. So the next thing we did was look into natural variation to see if these kinds of changes existed in nature, because we have lots of pathogens, of course. And so this is the DNA binding domain with positively charged residues indicated in red. And while there wasn't much variation over here, admittedly, this segment that I just pointed out, I was looking at this several pathogens, and this Cryptococcus gattii, which is a variant of Cryptococcus neoformans, it's particularly bad in the Pacific Northwest, had a couple of positively charged changes here, and so I simply made these mutations and asked how they affected invasion. And so this is what a wild type invaded cell or invaded spot looks like, with a washed off the surface of the plate. And if we make this Cryptococcus gattii mutation, we see that invasion isn't increased. And if we make a negatively charged change over in this helix, predicted helix anyway, we can get rid of invasion. And similarly, on the former side, if we make a somewhat synonymous change, positively charged change, invasion isn't so affected, but if we change DNA binding at this helix, that invasion seems to increase. And lastly, we also saw that if you put these mutations in a TEC1 deletion background, they're actually able to invade independently of TEC1, which makes us think that sterol 12 perhaps binds DNA slightly differently when it's heterodimerizing with TEC1, and these small biochemical changes have affected a similar change. And so that is that. So I've told you about this selection we can use to view this trait interaction through a set of small changes. There might be a trade-off in the domain, and some of these can act without TEC1. Sorry. Thanks.
So I might have missed this, but is there an evolutionary reason why both functions of sterol 12 should be in the same protein, or is there evolutionary pressure to duplicate and have paralogs with different functions?
Yeah, having looked in a lot of fungi, you can see that this pathway especially has been reappropriated in several ways. Like every now and then, sterol 12 will have some association with meiosis, or TEC1 might be involved in mating, and so there seems to be a lot of fluctuation in this pathway. I don't know of that many examples of a sterol 12 duplication event, but I should look at that sort of in more detail.
Just one technical question. Why do you have no data for some positions, and do you have strategies to get that information?
Yeah, the way we do mutagenesis is sort of a random doped oligo method, and so certain amino acid changes are almost impossible to get to from a certain DNA sequence. Tryptophans, for example, there's only one codon. It's hard to achieve. So we didn't specifically engineer those mutations, but I suppose there are other methods we could use to add those. Thanks.
Did you check a association between TEC1 and STAR12 with these mutants?
No, that's something we're working on now, though.